Good evening, ma'am.
Good evening, sir.
Good evening, ma'am. சொல்லுங்க மேம். Yes ma'am, was here ma'am. Good evening, my dear students. You are all are warm welcome to the another session of the biological classes. Today, what we are going to discuss, bioinformatics and their extents. Okay. So last class already we discussed what is meant by bioinformatics. Okay. Bioinformatics, plant as well as the animal, they are having the genetic material. The genetic material, what they are having, that things we analyzed, and also what is that? We have to store in the form of database. That is very, very important. See that one? The bioinformatics is an emerging field consisting of molecular biology and the computer science that enter on the development of the database, computer search algorithms, gene predictions, software, and other analytical tools that are used to make sense of DNA and RNA and protein sequences data. So what are the DNA molecule? It consists of already know about nucleotide. Okay. So nitrogen base, sugar molecules, and also phosphoric acid. These three included DNA molecules. So and also the sugar molecules also vary. DNA is a deoxyribonucleic acid, and the RNA contains ribonucleic acid. And what about the protein molecule? Already know about. There are 20 protein molecules also there. So they all are essential and non-essential. They are divided according to the requirement. Okay. And also the protein molecules is nothing but the more than one amino acid united together with the help of the polypeptide chain. Okay. These are all are the data analyzed from the different organisms and also stored in the form of what is a database in computer. So that is called as bioinformatics. Already we discussed in the last class. What are the bioinformatics? These are the other subject also included. So first one is computer is essential for the data analysis. Chemistry. What are the types of chemical substances present in the living organism? And biochemistry. So living organisms, what are the physiological functions occurs, and chemistry related one. And biology, already the science that deals study of the animal as well as the plant is collectively called as biology. Statistics, then only we may come to the conclusion. Statistics means it's nothing but the mathematical orientation, and also the mathematics as well as the statistics, these both are included. Okay, about the census also taking, isn't it? With the help of statistical department. So mathematics is small, calculation is there already but there are so many calculations also included in this part. And engineering means we can construct a new one. What is meant by engineer? They may construct a new building, isn't it? Likewise, bioinformatics also, they are having the genetic engineering. Genetic engineering means we can construct and each and every scientist, they can construct their own sequences. We can addition, deletion, like that. There are so many research also. Then after that, they will enter into the genetic engineering. We can construct our own gene also. Okay, so there is the things. Bioinformatics already we discussed. And also the bioinformatics also used for the pharmacogenomic. Pharmacogenomic is the study of how an individual genetic inheritance affect the body response to drug. This is very, very important. Okay. The terms comes from the word pharmacology and genomic, and it is thus the intersection of the pharmaceutical and genetics. These two are interrelated. Then the pharmacogenomic is possible. So what is the use of this one? Already we discussed these things also. For example, a particular person, he has a own specific DNA sequences. Okay, this DNA sequences or genome, it is resistant to the particular drugs. Okay, each one has specific resistant capacity. Okay, that's things also we were analyzed. We will analyze. So this is called as what is that? Pharmacogenic. In this category, see that first persons, they need only one tablet for their disease or restriction for the microorganisms. Second person of tablet is enough. And third person, they need two. The last one, they do not need even a single tablet. So these are all how they analyze the means. These are all present in your gene profile. Okay, it is also derived from the one generation to another generation. That is very, very important, my dear students. Okay, so what are the beneficial for pharmacogenomic? Already we discussed. So more powerful medicine, how will you identify it? Means the particular person, they will not accept the medicines, means we have to give more amount. That is called as overdosage. Take an example, so even single infants or fetus or children, they will not take more dosage, isn't it? Suppose take an example, some of the persons, they need more powerful medicines. Okay, then even take an example of ml or mg, like that. Age group is also there. Depends upon the age groups, the medicines also prepare. We will not provide the infant tablets, isn't it? We will provide the liquid medicines. Okay, syrup, isn't it? Simply called as syrups. That is the first one. And the second one is between the safer drugs, the first times. This is also another one. Pharmacogenomic, which one is the safer for your body? Which one is accepted in your body? That things, how can you identify with the help of the pharmacogenomic only? And third one is advanced screening for the diseases. And one more is that what are the diseases it will come in future? We can identify it from the genome. That is the third one. So then the last one is also better vaccination. Which one is easily accepted in your body? Which one is not accepted in your body with the help of bioinformatics or pharmacogenomic? We can easily identify it. Okay. So these are the 4 points is very, very important, my dear students. Go to the next one is 21. See that one? This is called as slide is very, very important. The simple drugs they used. 3 categories have been reported here. One is a slow metabolizer, because the absorption capacity and the ability and the age group, they are all also very, very important in this field. And second one is normal metabolizer. Normal metabolism can be easily absorbed 50% only. The rapid metabolism, within a short period, 2 minutes, 2 hours or 1 hour, the absorption of tablets takes place within the body. These are the 3 categories have been reported according to the pharmacogenomic. Okay, next one is a very, very important, my dear student. This is very, very important. Don't forget, my dear students, this is the DNA fingerprinting. So here, what we are going to discuss about, so what is by DNA structure already we know about. We have to find out the DNA fingerprinting of the culprit. There are so many technological methodology also used for the DNA fingerprinting. Exact copy of the DNA molecules we can easily identify from the DNA to cellophane paper. Okay, what is my fingerprint? Finger, when you are getting the each one, it has a specific finger line is there. Okay. Likewise, DNA fingerprinting also we can take. So it's also one of the techniques. See that who discovered first? What are the important. What are the technique? How will you identify the DNA molecule? How will you separate the molecule? And then how it will be separated in the form of what is that? Nucleotide, and then how it will be entered into the database. Okay, that things we are going to see. Please watch carefully. So DNA fingerprintings. So the technique of the DNA fingerprinting was pioneered and perfected by British genetics. The name of the scientist, Dr. Alex Jeffreys. Don't forget, my dear students. British genetics, Dr. Alex Jeffreys. Second one is DNA fingerprinting is a quick way to compare the DNA sequences of any two individuals. Take an examples, father and mother, okay, and father and children, and also culprits, okay, any malpractice, murder, whatever it may be, okay, suicide, whatever it may be, we can easily find out the sequences, whether it may be similar or dissimilar, who is responsible for that particular incident or something else, whatever it may be. See that one, it involves the identify the differences in some specific region in a DNA sequences called as repetitive DNA. Already we told. Okay, the repetitive DNA, how many base pair is present in it? 1.2% is single base pair. Already we studied. 50% of the base pair is what is that? Repeated, otherwise called as junk DNA. Okay. It is also present in the both the characters. Those, suppose take an example, you are the son, isn't it? Your father is also having a DNA molecule. Isolate the DNA molecule and analyze it. What will happen? Your DNA molecules as well as your father DNA molecules is similar in nature. Okay, so that is also called as what is that? Repetitive DNA. Because in these sequences, a small stretch of the DNA is repeated many times. That is called as repeat. Small stretch of the DNA is repeated again and again. How many times we cannot be able to calculate each and every one. It has a specific, what is that? Properties. Okay, so that is also very, very important. So go to the next one. So DNA fingerprinting definition. What is the exact definition of the DNA fingerprinting? See that DNA fingerprinting is a laboratory technique used to determine the probable identify of a person based on the nucleotide sequences only of certain region of the human DNA that are unique to individual. Suppose if you are already told you, take an example, see any two individual, whether it may be a same family or different family. We can analyze it. Okay. You and your brother. Okay. You can analyze you and your sisters, whatever it may be. You can analyze it, the nucleotide sequences. You can see the certain region of the human DNA that are unique to individuals. Okay, you and your brother, your sister DNA will be matched 99.9% only matching. 0.1% will be differ. Already we discussed in the previous slides. So that is the exact definition of the DNA fingerprinting. Okay, once again, DNA fingerprinting is a laboratory technique used to determine the probability, identify of the person based on the nucleotide sequences of the certain regions. Certain region means any one of the location. Take an example of 5, 10, 20, like that. That region of the human DNA that are unique to individuals. Each and every one, they are having the unique DNA sequence. So that is very, very important, my dear students. It will not match you and okay, your neighbor. That is very, very important. Just like your fingerprint. Your fingerprint is not similar to that of the your brother. Isn't this? So that is very, very important. What the next one is, see here, this is the steps involved the fingerprinting technique. What are the steps they were followed in the laboratory aspects? How it will be identified, the DNA finger? How did you isolate the DNA from the samples? That things you must note it on. These are the 7 steps they were followed. Some of them are having 7, some of them are having 10 like that. But I have chosen here 7 steps. First of all, you should collect the DNA samples. So, okay, you have to collect the DNA samples. What is the meaning of DNA samples? You can take any one of the tissues or blood samples. Okay, so these are all are very, very serum, even serum. Blood is the main one because it's contained the nucleic acids, and tissues also it contains DNA samples. It may be liquid part or solid part from the person or any object, living object. Then from the DNA sample, after the collection of DNA sample, you have to isolate the extraction of the DNA molecule. Particularly, you have to isolate the DNA from the cells. There are so many processes which occurs within these two: collection of sample and the extraction of the DNA molecule is also very, very important. After that, cleavage of DNA by restriction endonucleases. You have to split the molecules into simple pieces or fragmentations. Okay, we cannot be able to find out all the entire sequences. Okay, we have to split the molecule into 200 base, 300 base pair. Then only we can easily identify it. Okay, that is very, very important. Third one. How will you isolate? Means the gel electrophoresis to separate the DNA fragments. This is very, very important. DNA fragmentation was done by the one of the most important technique is called the gel electrophoresis. The electrophoresis is nothing but the instrument used for the separation of the DNA molecule. This is very, very important in the form of fragments. Fragments means already told you a small polypeptide chains. So here the technique they were used is southern plating technique. So many things is there. Southern plating technique is also there, and western plat, northern, eastern, whatever it may be, because depends upon their country position. Southern plating techniques they were used. After that, autoradiography to obtain the DNA bands. It's also connected with the computer. Okay, the result will be comes out in the form of autoradiography to obtain the DNA band. I will show you what is meant by band. Okay, band means it's nothing but small. What is that? Already studied in the myosins, muscle structures, light band and also dark band. Okay, likewise, depends upon their concentration of the substance. So likewise, here also we will obtain the band where in the autoradiography to obtain the DNA band also, whether it may be concentrated or a mild one, like this, depends upon their number of nucleotides. And the last one is develop and visualize the DNA fingerprints. How will you visualize the DNA fingerprint? After you will obtain the DNA molecules or DNA fingerprint is how they will be obtained. These are the steps that were used for the identifications of the fingerprinting technique. So this is the band. See that this is the band. Okay. So this is the band. See, this is the place already in our lab. They have placed the molecule samples. Okay. So it is also kept under the electric field. As a result, what happened now? The molecules contain positive charge as well as the negative charge. They can move from the positive charge, they can migrate towards the electric field in minus, and minus means that negative charge, they can migrate toward the positive, that is anode. As a result, you will get it the, what is that? Band-like structure here. So this is also one of the band. Okay, this is also one band here. So likewise, it may be. So you will get the unequal size of band structures. The concentration depends upon the molecules only. This is very, very important, my dear students. Okay, so DNA fingerprint. So after the electric field, after the gel electrophoresis will obtain the DNA structure will be like this. Okay, so see here. This repetitive DNA are separated from the bulk genomic DNA as different peaks during density gradient centrifugation. So this is the method, density gradient centrifugation method. This is the first step. After the collect of samples, we will put into the, we have to add some amount of the chemical substances also there, and also grain well, and also kept under the centrifugation process. You will get the solution that is called a supernatant. Another one is called as what is that? Bottom there is a high molecular weight salt deposited on the bottom of the test tube. Okay, the bulk DNA from the major peak and the other small peaks are referred to as the satellite DNA. Here you must note it down. Two types of peak we will get it when you are getting the centrifugation process. Okay, the bulk DNA forms a major peak and the other small peaks are referred to as the satellite DNA media. See here, this depends on the base composition. Already we know about DNA containing the 4 nitrogen bases is there. One is called as adenine, second one is thymine, and rich or GC rich also. These are the two things is there. 4 nitrogen bases already we studied, purine and pyrimidine. The length of the segment and the number of the repetitive unit satellite. So the satellite DNA is nothing but what is that? The composition of the nitrogen bases. Okay. So this is also classified in the DNA is also classified into many categories such as the macro satellite and microsatellite and mini satellite according to their number of nucleotide. Which one? The number of nucleotide is nothing but these things: adenine, thymine, guanine, cytosine, otherwise called as purine and pyrimidine. Simply we may call as nitrogen bases. According to these things, they may be divided into 3 things is there. One is microsatellite. Micro means small one. Mini means medium one. Macro means more one. Okay, so etc. They have been reported. See the next one. These sequences normally do not code for any protein, but they form a large portion of human genomes. This is a very, very important one. Okay, these sequences normally do not code for any proteins, but they form a large portion of the human genome. This is very, very important. The human genome contains, there are so many sequences that sequences particularly mention the protein molecules. Some of the sequences, they will not mention the protein molecules, any code. So that has given here. Okay. These sequences show a high degree of polymorphism. Polymorphism means, more than one same structure and form the basis of DNA fingerprinting. These are all very, very important, my dear students. So these are all very, very important. Once again, repeat for you. Okay. So what are the steps involved in this? DNA samples, collection of DNA samples, and the extraction of the DNA sample from extraction of the DNA from the sample, and cleavage of DNA by restriction endonuclease. You have to get the simple fragment like structure. After that, it will be applied into the gel electrophoresis to separate the DNA fragments. After that, southern plating technique, they were used for the separation of molecule and also used as a autoradiography to obtain the RNA as a band. After that, it will be visualized DNA as a fingerprinting. Exact copy of the DNA, we can easily visible in nature. We have to use some chemical substances, then only it will be visible in nature. These are the band, already we saw it. How it will be, already it depends upon the adenine, thymine, guanine, cytosine. They are all classified into 3 categories. One is called as microsatellite, another one is called as macrosatellite, and third one is called as mini satellite, etc. Okay, so the mini satellite, what is the meaning of mini satellite? The mini satellite were first found in the human genome and described as hypervariable tandem repeats with monomers, repeat length of the about 10 to 100 base pair of more than microsatellite. That means the mini satellite, okay, it contains 10 to 100 base pair of microsatellite. So it is also very, very microsatellites more than 100 base pair present in it. Okay, how many times they repeated the length again and again and again? I already told you 1 to 10, 1 to 10, 1 to 10. So this way, how many times it repeated? 100 base pair is also repeated. We may come to the conclusion: microsatellites are united together form a mini satellite. Okay, repeated several times, less than microsatellites. So this is also very, very important. A number of repeated unit in the loci, otherwise called as location, the position, varies between the genotype and it is referred to as the variable number of tandem repeats. This is called as VTNRS. Sorry, BNTRS. This is very, very important, my students. So many times they were asked the question in mnemonics or expansible. Okay, what is my VNTRS? VNTRS is nothing but the variable number of tandem repeats, or the hypervariable region, that is also denoted as HVRs. Hypervariable and also a variable number of tandem repeats. Repeats, I mean repeated again and again. Already I told you. Repeated AUG like that. Variable means it may be different. Okay, it is not repeated. Okay, such type of comments. For example, one is AUG, another one is GUA. Next one is UGA. Next one is GUA. Okay, likewise, it may be variables. So there are two types of variable have been reported. This is also very, very important, my dear students. See the structures. This is a mini satellite. This is called as a mini satellite structures. Already told you, mini satellites are composed of 100 base of the micro satellite. Okay, so this is called as mini satellite. Don't forget, my dear student, this is also a exact structure of the chromosomes. Which one? Mini satellite. Next, we will pass on to the next slide is called as mini satellite and micro satellite, and also that macro satellite, how it will be divided, differentiated. See that this is the exact chromosome, normal chromosome, whether you take an example of X chromosome or Y chromosomes. You can imagine chromosome. The chromosome it contain genome, genome contain gene. The genes, it depends upon the gene structure. It is also depends upon their nitrogen bases, adenine, thymine, guanine, cytosine. It is constructed by the DNA molecule. 3 components there. Nucleotide, nitrogen base, sugar molecule, and phosphoric acids. They are all collectively called as nucleotide. They may be divided into 3 categories according to their base pair only. Already I told you. What is my microsatellite? Microsatellite maximum consists of 1 to 9 base pair. This is very, very important, my dear students. Microsatellite 1 to 9 base pair. And what about the mini satellites? Mini satellite 10 to 100 base pair. Mini satellites also 10 to 100 base pair. Okay. So this satellites or mini satellite, micro satellites means what is that? Mini satellite. Micro, more than 100 micro satellite united together forms a mini satellite. Likewise, see that more than 100 mini satellites get together form as a macro satellites. Okay, so there are 3 types of chromosome present in one: micro satellite, mini satellite, and macro satellite. Each one interrelated. So these are all TR as tandem repeateds. Okay. These 2 are all called as what is a tandem repeateds, but this one is not, comes under the heading. So this is also very, very important, my dear. So microsatellite, already we discussed. Microsatellite also known as the short tandem repeats, microsat, simply called as STRS. Okay, microsatellite is also called as a short terms repeats or symbol sequence repeats, SS or STRS, or short repeating nucleotide maximum. How many nucleotides mostly present? 2 to 6 nucleotides also present in it. That's why it is called as microsatellite. Micro means minimum 2 to 6 nucleotides present in it. Okay. In plant, di, tri, and tetranucleotides repeat are found widely distributed. Dinucleotide, trinucleotide, tetranucleotides. So what is the meaning of nucleotide? Di means 2, tri means 3, tetra means 4 nucleotides. Repeated are found widely distributed in plant. Don't forget, my dear students. Sometimes they may ask the question in this part. Dinucleotide of the microsatellite mostly found in plant, animals, and particularly they may give any one of the examples. Trinucleotide also. So you should note it on this one. Di and tri and tetranucleotide repeated or found widely distributed in the plant only. And the next one is the copy number of these repeats varies from individual to individual and form the basis of polymorphisms. The structures also is not similar in plant. Di or tri or tetra, the copy number of these repeats vary from individual to individual. For example, banana is there, and also mango is there, tomato, potato, whatever it may be. Each one has a own nucleic structures, but it is not similar. It has a own, what is a repeated sequences. There varies, differences present in it, individual to individual, and forms the basic polymorphism. This is a very, very important one. See the structure. So this is the structure is called as microsatellite. See, this is a chromosome. This is collective called as satellite DNA. It is a nucleotide, already know about. It composed of the mini satellite, microsatellite, and microsatellite. See that this is the structure of microsatellite. The particular one square, the one square denoted here, no, this is considered as a mini satellite. more than one. From this one, there is a microsatellite. This is the four category, four nucleotide present, and this is called as what is a microsatellite. Okay. So the microsatellite more than 2 to 6 microsatellite, 100 microsatellite united together forms a mini satellite. So in this portion, okay, you can easily identify this spot. So don't forget, my dear students, how it will be mini satellite and microsatellite. What is my mini satellite? See here, just you should understand. So this is the part. So this is the DNA sequences. Taken examples. This is considered as a DNA, isn't it? DNA. From this portion, we are cutting from this one. You have to cut this fragment. Okay, this fragment containing what is that? So the particular fragment is like this. You have to cut this portion. Cut the portion. This part. Here, mini satellite. What is the meaning of mini satellite? What is the meaning of microsatellite? Mini means 4. For example, this part only. Here divided into so many things. 1, 2, 3, 4, and 5. This is called as what is a mini satellite. Mini, okay, satellite. Okay, mini satellite. Okay, see that one, mini satellite. Here, what happened? See here, there are so many nucleotide present here. How many nucleotide is there? See that different nucleotide also present. See, for example, so A, like that. This portion is called as what is a mini satellite. Okay, for example, only one section. This one section is called as what is that? Mini satellite. This one is also another mini satellite. So the mini satellite contains what is a nucleotide. This mini satellite united together form as a microsatellite. This united, this part only, what happened, just imagine. Okay, the AAA, AUG, likewise it will be presented. So more than one mini satellite united together forms a microsatellite. These are the mini satellite. This is called as one mini satellite. This one second mini satellite, third and fourth. This mini satellite united together forms a which one? Microsatellite. Okay, this is very, very important. Microsatellite. Okay, satellite. This is very, very important, my dear. Okay, so this is very, very important. So you have to find out each and every DNA molecule is composed of the microsatellite and as well as mini satellite also. The microsatellite is nothing but the combination of adenine, guanine, thymine, cytosine, the components. So each microsatellite united together forms a mini satellite. And micro and macro united together forms a DNA structure. That's all about, my dear. Okay, so we will see the next one is, see that one, this portion that is given here. See that this is mini satellite. This part is mini satellite. Okay, it contains the mini satellite is also denoted as a nucleotide. Each and every nucleotides united together forms a microsatellites. So collectively called as satellite DNA. That's all. Okay, the satellite present in it, that is called a satellite DNA. Some of them are non-satellite DNA is also present in it. This is very, very important. Okay. Next one, we'll pass on to the next one is called as what is the principles of DNA fingerprinting. What are the principles they are following? How it will be isolated? The things we are going to discuss, my dear students. First, principles of the DNA fingerprinting. How it will be? What are the main principles? Short nucleotide repeat that vary in number from person to person, but are inherited or variable number of Denton's repeats sequences. Already we discussed this part. Okay, so this part, what is that? Already we discussed, isn't it? What is that? What is the meaning of this one? You can see it. Number will be very repeated components, repeated DNA sequences. Person to person, it will be vary. They are collectively called as a VNTRS. That things we are going to analyze from the fingerprinting. Then only we can easily identify the person, whether related or not related. The first things. And second things, the VNTRS belong to class of satellite DNA referred as the mini satellites. This is also satellite DNA is nothing but the VNTRS. That is called as variable number of tendons repeats, united or sequences present in the DNA chain. So then we'll find out the mini satellite. What is the meaning of mini satellite? Already we discussed. A small fragment, a segment. Then only the mini satellite, it contain adenine, thymine, guanine. So number of adenine, number of guanine, number of thymines, how many number present in it, that can be referred to the another one. Then only we can find out who is culprit, who is relative, who is the relations. Like that we can easily identify. How they are identified? With the help of the nucleotide base pair only. The adenine, guanine, thymine, cytosine, each and every position, each and every mini satellite only we identified. Then only we can find out the malpractice persons. Both the characters, both the persons, they analyze the mini satellites from the DNA molecules. It differs from the individual to individual in a populations, except in the case of monozygotic identical twins. You cannot be able to find out which one is differ, but particularly 0.1% will be differ, but we cannot be able to identify it except monozygotic. Monozygote means same zygote divided into two when during the morula blastula stage. It may be divided. It's a single zygote divided into two. And also two again, what happened? It split into two, and the two undergo the developing process. The two identical one. So that is called as monozygotic. Okay. And the VNTRS of the two persons may be of the same length of the sequence at certain sites, but vary at others. This is very, very important. These are the principles of the DNA fingerprinting techniques. So next we will pass on to the next one is called as, what is it? The child might inherit a chromosomes with a 6 tandem repeats from the mother. Note it down. This is very, very important. How many tandems is the 6 tandems from the mother and 4 times tandem from the father. So this is very, very important. Sometimes they may ask the questions. The child might inherit a chromosomes 6 tandems, 4 tandems, 5 tandems, 2 tandems from the mother. Sometimes they may ask the question from the father. Suppose what will be the ratio between like that, father and mother? Like that they may ask the questions. Okay. It is also very, very important, my dear students. So mother 6 tandems, father 4 times. See that one? The child might inherit the chromosomes with a 6 tandems repeat from the mother, and the same tandems repeat 4 times in the homologous chromosome inherited from the father. This is very, very important. 4 times. Okay. 4 times. Don't forget, my dear. This is very, very important. Father and mother. Okay. Father how much? 4 times. Mother how much? 6 times. Okay. So this is very, very important. And then half of the VNTR alleles of the child resemble that of the mother and half that of the father. Okay. Maximum it is related to the which one? Mother only. From the half means what is that? 50% of the father, 50% from father or mother, remaining 50% will be father. Okay. Yes. So that is very, very important, my dear students. Okay. So this is a very important things. And go to the next one is, we'll pass on to the next one is question part. Okay. I think that the part will be completely finished, I think. Okay. See this one. Question part. From these questions, you must note it down. You have to pick out the correct answers. Which one is correct statement given here? Which of the following is a correct statement in the human genome? The largest known chromosome gene is dystrophin at 2.4 million base pair. Okay. This is a a correct one. Already we discussed which one is the highest one. Dystrophin, highest or largest known human genome is dystrophin 2.4. Already we discussed, it's a correct one, correct. And second, about 10% of genome is at telomere and around centromere. Already this one is also correct. Already we discussed in previous slides. And microsatellites, also known as short-term repeated STRS and also symbol sequences repeated SSRR, short repeated in nucleus 2 to 6. Now only we studied this point, also 3. This one is also correct. Okay. The 4 chromosomes, one has almost genes 2968. Already last class we discussed this part. And the Y chromosome has the fewest 231. So the all are correct. Only is the answer. Isn't it? All are correct. Because already know that dystrophin is the largest known human genome. 2.4 million base pair. Yes. 10% of genome is telomere. Already we discussed that things also yesterday. Microsatellite, also called a short tandem repeats or symbol sequence repeats. It contains the nucleated 2 to 6 nucleotides. Already discussed. Now only we studied it. And chromosome 1 has the most genes. Yesterday we saw it 2968, and also Y chromosome has the fewest 231 nucleotides. So the answer is which one, you know? See that statement A and B correct, statement B and D correct, statement A, B, C are correct, statement all are correct. So the answer is which one, you know? The 4th one is the correct one. Which one is the correct one, you know? The 4th one is the correct one. So we have to find out the answer from the questions. So which one is correct? Carefully you have to read the questions. These are all are considered known as statement type of questions. Okay. So what the next one is, satellite DNA is important because it, yes, the satellite DNA is very, very important. Why it is? The code for enzymes need for DNA replication. Is it correct? Is it correct? No. Okay. So DNA replication process do not occur here. Code for protein needed in cell cycle? No need. Okay. Protein biosynthesis, there is no need of code. Genetic code, no need. Okay. For protein needed in cell cycle, cell cycles, what is that? Protein synthesis means it's okay. That one is so high degree of polymorphism in population and also same degree of polymorphism in individual, which is inheritable for parent to children. This is exactly correct. What about the last one? Does not code for protein and in the same in all member of the population. So the first one, second one, the last one is not correct one. The third one is the correct one. How it will be possible? Already we discussed these things also. The character will be transferred from the parent to children. It's a correct one. Okay. The polymorphism, high degree of polymorphism in population, and also same degree of a polymorphism is an individual which is inheritable. Those who are having individual polymorphic characters from the mother or father, it may be transferred from the parent to children only. So that one is correct. So these are all are what is the high polymorphism where it is present the satellite DNA. The satellite DNA contain many polymorphic mini or micro satellite DNA present in it, isn't it? That's why the third one is the correct one. Okay, see that one? The correct, so the high degree of polymorphism in population and also the same degree of the polymorphism is an individuals, which is inheritable for the parent to children. This is the correct way. Okay, so go to the next question. The mRNA directs the building of proteins through a sequence of, which one you know? The building of proteins. Protein molecules mean which one they will essentially? Exon? No. Introns? No. Codon is very, very important because the codon mostly present in where you know? mRNA. Don't forget, my dear. mRNA. Okay. So it is very, very important one. Anticodon, it's present the where you know? tRNA. This is the complementary of this one. Okay, here what they are asking, the mRNA only they ask, isn't it? mRNA means which one is responsible? The codon only the responsible. So the answer is C only. Okay, this is the anticodon. It is a complement of which one you know? mRNA sequences. What the mRNA sequences consist of? It is complementary to the tRNA molecule. Already we studied in the protein biosynthesis or translation process. So here the answer is which one? Code mRNA only they mentioned. So the exon is not comes. Exon intron mostly present the DNA molecule. Anticodon tRNA. So mRNA means which one? Codon is the responsible because it has a specific message from the nucleus or genetic code from the model. That's why it's a codon. Okay. Go to the fourth one. The transcription enzymes present in the nucleolus is involved in the synthesis of which one? Normally. So there are so many rRNA molecules is very, very important. Here what they are asking, the transcription enzymes present in the nucleolus is involved in the synthesis of 5S rRNA and 5S rRNA and Hn RNA. It is not. And second one is already here given as answer. B was the correct ones because 28S RNA and 18S rRNA. So this is the correct one. The 28S. Sn RNA, Hn RNA, it will not come. So only this part one is the correct one. This is the correct answer only. These are all direct questions. These are all direct question asked by the NEET. And last but not least, the last questions. Just imagine this is very, very important. Already we saw it. This is the direct questions. Which of the following statement is correct? How many nucleotides in the human genomes? This is the questions. Okay, this is the questions already know all about. Among this one, just note it down. The first one, just listen here. So 3,164.7 million chemical nucleotide. This is the correct answer, isn't it? So remaining things are, I think it is not a correct one. So you have to find out the answer, how it will be. The number will be similar in nature. Here 3,164. Here 3,146. Okay. Don't confused. Okay. 3,164.7 million chemical nucleotide base pair is present in the human genome. Others are wrong only. See that 3146, here 1346, here 4316. So these are all wrong. 3164.7 million chemicals nucleotide base pair present in the human genomes only. And sometimes they may ask the question, which one is the largest genome? Dystrophin, how many nucleotide present in it? Like that, they may ask the questions. Okay. So this is the last question, my dear students. That's all about the part 10, molecular basis of inheritance. The part 10 will be finished. We will pass on to the next one is part 11. So before that, we may summarize. Next class, we will see. Before that, we summarize all those things once again. Okay. Last class, what we have discussed, bioinformatics. Today, this class, we will discuss. We were discussed DNA fingerprinting. In this part, DNA fingerprinting, the technique of the DNA fingerprinting was pioneered and perfected by British genetics. Don't forget, my dear students, Dr. Alex and Jeffries. Alex and Jeffries. This is very, very important, my dear student. This is very, very important. Alex and Jeffries. Okay. So this is an important point. So many times they were asked the question, DNA fingerprinting was discovered by the name of the scientist. Sometimes they may ask the questions, their country also. And second one is DNA fingerprinting is a quick way to compare to the DNA sequence of any individual. You can easily identify it. Okay. From your brother or your sister or cousin sister, whatever it may be. Some of the malpractice, murder, forensic. So all those things we can easily find out the answer.
Someone is called.
Next step. And pass on to the next one is DNA fingerprinting. It's already in our world, but who discovered and how it will be? That things also we'll discuss. What is the exact definition of the DNA fingerprinting definitions? DNA fingerprinting is a laboratory technique used to determine the probable identify of the person based on the nucleotide sequences of a certain region of the human DNA that are unique to individuals. Okay, so this is a very, very important one. Already, what are the steps involved in the DNA sequences? See, the steps involved in the DNA sequences already we discussed. First, collect the samples, and from the collect samples, we have to collect the DNA samples also. And also after that, the extraction of the DNA molecules and cleave the DNA molecule with the help of the enzyme is called as stress section endonucleases. That's why endo means inside, cutting inside the nucleus. That's why it is called as endonucleases. After that, the fragmentations also run into the gel electrophoresis to separate the DNA fragments according to their molecular weight and also that component or content. So which method? There were southern plotting techniques they were used for the fingerprinting technique, DNA fingerprinting technique, and the audio radiographics to obtain DNA bands. So this is also very, very important. Last one, what happened? Automatically we will get the DNA fingerprint, okay, in the visual aspects. These are the steps, my dear students. Don't forget. You must note it down. 2 points is very, very important. Which method they were used? Southern plotting technique. Okay, that method. And which instrument? Gel electrophoresis to separate the DNA fragments. That is also very, very important, my dear students. Okay. So then after that, samples, how it will be, look like a band. So, okay, light band and the dark band is also obtained with the help of, what is that? Gel electrophoresis. So the gel electrophoresis, we will get it this type of the structure according to their molecular weight. The DNA molecules also separated in the electric field. And then, so we'll pass on the next week. Next one, these repetitive DNA are separate from the bulk genome DNA as different peaks during the density gradient centrifications. During the centrifugation process, we will obtain 2 types of peak is there. One is a bulk genome DNA peak, and also another one is called a small peaks also there. We will obtain. The bulk DNA from the major peak and the small peaks are referred to as the satellite. Which one is the small one? They are all are concept that is known as satellite DNA. The satellite DNA consists of what is that, about the depending on the base of the composition, adenine, thymine, and also rich or guanine, cytosine rich. The length of the segment and the number of the repetitive unit satellites. Okay, DNA are classified into many categories such as microsatellite and mini satellite. We discuss. And the next one is these sequences normally do not code for any protein molecules, but they form a large portion of the human genome. So it will not code for any protein molecule, but it present inside the genomes only. It has a large amount of genome present in it. These sequences show a high degree of polymorphism and form the basis of the DNA fingerprinting also. Okay, these are all very, very important, my dear. And mini satellite, what is my mini satellite? How it will be constructed by the nucleic side. The mini satellite were first found in the human genome and described as hypervariable tandem. This is very, very important. Hypervariable tandem. Repeat with the monomers, repeat the length of the about 10 to 100 base pairs more than microsatellite. Repeat several times less than microsatellites. This number of repeated units, the loci varies between the genotype, and it's also referred to as the variable number of tandems repeat. This is very, very important, my dear students. Simply called as VNTRS or the hypervariable region. Both variable region present in the mini satellites and repeated again and again. So, okay, the structure look like this one already all about. Okay, the exact structure is also this one. So inside what happened is DNA is the DNA inside where it's a mini satellite and microsatellite is also present in it. And mini satellite, last one is this is the structure, entire structures. Mini satellite, what is my microsatellite, what is my TRDS, all those things it may be mentioned in the single slide. So this is a chromosome. Inside there is a genome is the inside the genome there is a DNA molecule is the DNA molecule consists of these are the 3 components. One is called as microsatellite, another one is called as mini satellite, and third one is called as macrosatellite. It has a specific base pair sequence, so 1 to 9 base pair in the microsatellite and 100 base pair in the mini satellite. Macrosatellite means these 2 united together form as a macrosatellite. This is also more than 100 base pair. So these are all are what is that TRS tandems repeated again and again. Microsatellite, mini satellite, macrosatellite. Again microsatellite, mini satellite, macrosatellite. Likewise, it may be arranged in DNA molecules. Okay. Microsatellite. It is also similar. How many nucleotide present in 2 to 6? Already we discussed. And this is the entire structure of the satellite molecules, and also mini satellite, how it will be forms, and also another microsatellite, and also macrosatellites also. What is the principles of DNA fingerprinting? Already we discussed. Okay. It is similar. Which one you know? It differs from the individual to individual in the population, except in the case of monozygotic identical twins. Okay. So that things also we will discuss. And children's, it is similar to that of the 6 tandems repeated from the mother and 4 times of the father. How many percentage? 50% from the mother, 50% from the father. Okay. And also remaining thing is we will see in the next class, my dear student. Thank you so much for your participation. Thank you, my dear. Thank you. Bye. Take care.